Hi, everyone. Good evening. This is Perspectives, a thought lead initiative by Kivani. Today, we'll be talking about COVID-19. Yes, a lot has been spoken about COVID-19, but today we are going to have a fresh perspective and a look into COVID-19. The reason is there is a lot of incorrect information regarding the virus. Knowing the facts grounded in the latest scientific evidence is the key to being properly prepared and protect ourselves. Help us understand the current situation, what are the important mutations that are taking place, and probably the topic which people are quite keen on in terms of the approved vaccines. We have Dr. Najla with us today. Dr. Najla is the head of Princess Dr. Najla bin Saud Al Saud Center for Distinguished Research in Biotechnology at King Abdulaziz University in the Kingdom of Saudi Arabia. Dr. Najla also serves as an associate professor at Biological Sciences Department, and she received her PhD in molecular biotechnology and has published many scientific research articles in the field. Dr. Najla has a patent entitled A Diagnostic Set for Detecting Avian Influenza Virus and teaches courses for undergraduates, postgraduates, and PhD students. So I think given her background and credentials, Dr. Najla can give a very keen and a scientific perspective into COVID-19. Before I turn the stage to Dr. Najla, just a little housekeeping. Please stay on mute and don't raise your hands during the presentation. If you have any questions in your mind during the presentation, type them into the Q&A box. You can see it in your Zoom control panel. Once the presentation is over, during the dedicated Q&A session, I'll bring up your questions and put it across to Dr. Najla. Now let's turn our attention to the main part of the presentation to know more about the virus. Dr. Najla, thank you very much for joining. It's a great pleasure to have you with us today. We are absolutely eager to listen to what you have to say. So I'm turning it over to you now.
Thank you very much. بسم الله الرحمن الرحيم وما تشاءون إلا أن يشاء الله رب العالمين. السلام عليكم ورحمة الله وبركاته. Good afternoon, ladies and gentlemen. My name is Najla bin Saud, and it's my pleasure to welcome you all. Today, I will participate and share a talk about a topic that occupies the first place in the headlines, COVID-19 disease.
Dr. Najla, sorry to interrupt.
Yes.
You have to come closer to the microphone, please.
Okay.
Yeah. Now it's better.
Yeah. Okay. Today I will participate and share in a talk about a topic that occupies the first place in the headlines, COVID-19 disease. The current webinar presents the available information about COVID-19, key facts, important mutations, and approved vaccines, which might help us understanding that epidemic to protect ourselves, families, children, and beloved ones. Now we are starting with a question. What are the coronaviruses? Coronaviruses are RNA virus that cause diseases in mammals and birds. The name coronavirus is derived from the Latin word corona, meaning crown. As we noticed from its shape. Here in the photo, we can see the spike protein from the top to RNA genome and the envelope and the lipid membrane and the other proteins in this picture. They are a large family of viruses that causes illness ranging from the common cold to severe diseases, such as Middle East respiratory syndrome, MERS-CoV, and severe acute respiratory syndrome, SARS-CoV. SARS-CoV-2 is the virus that causes COVID-19 disease. On March 11, 2020, the World Health Organization characterized COVID-19 as a pandemic. SARS-CoV is not a new virus, by the way, as someone thought. According to WHO, it was first identified at the end of February 23 during an outbreak that emerged in China and spread to 4 other countries. So, what are the differences between SARS-CoV 2003 and SARS-CoV 2019? Both belong to the genus beta coronavirus viruses, lineage B. According to WHO, the fatality rate of 2003 is 9.6%, and it's about 3.9% in 2019. In conclusion, the difference in SARS-CoV-2 2019 is the emergence of a new stem loop. As we see, the nucleotide sequence of 5' untranslated region UTR of 2019 SARS-CoV-2, which represent the formation of SL4', new stem loop, and C2U mutation in SL5 stem loop. To protect ourselves from the infection, we have to understand some important facts, such as how does the virus enter our cells and cause infections? As we see in this figure shows the life cycle of coronaviruses to enter the cell and cause infection. The virus has attached to the cell receptors named ACE2 in the case of COVID-19 and SARS-CoV-2, and named DBB4 in the case of MERS-CoV. Then it entered the cell by endocytosis. After that, the virus releases its RNA and begins the replication, transcription, and translation to form a huge number of new viral particles, which released from the cell by budding to infect a new cells. I think it's very clear in the picture. It gives you an idea, a clear idea about what's going on. As we all know, there are 3 main locations for coronaviruses to enter a human body: mouth, nose, and eyes. And I think everybody, he knows already about this one, but we have to mention it because it's very important how to take care. The time between exposure to COVID-19 and when symptoms start, which named incubation period, it's commonly around 5 to 6 days and ranges from 1 to 14 days. And it's very important to know that the incubation period for children is the same as in adults. Now we have a case study about a man named Sam. For Sam, at day 0, Sam was exposed to COVID-19. At day 5, he was tested for COVID-19, and the result comes back negative. by day 8, thinking he did not have COVID-19. So he went to a small gathering. He was contagious. On day 8 and 9, 24 hours before symptoms, and exposed to 10 people. At day 10, he became symptomatic and tested positive for COVID-19. Here we have to mention that we have to really take care, because I know a case who tested positive for COVID-19 many times for a long, long time without any symptoms at all. So you have to take care, all of you, because sometimes the symptom doesn't appear quickly. It's well known that the most common symptoms regarding to COVID-19 disease are alveolar edema, difficulty in breathing, and inflammation due to the lung infection. Though the lung is the most effective organ, we have to know that other organs can be affected as well, such as intestines, brain, heart, vessels, and kidneys, causing severe symptoms. As if the breathing complications associated with COVID-19 aren't worrisome enough, doctors are discovering blood clots as another risk posed by the coronavirus that can lead to life-threatening, like strokes, heart attacks, and pulmonary embolism. And here we try, I try my best, and we have to understand this is not the only reason, but here we are trying to explain the formation of mysterious clots in COVID-19 patients. Patients with COVID-19 are at significantly increased risk for the formation of blood clots. The pathogenesis for their development is incompletely understood. One possible explanation is as follows. The binding of the spike protein of SARS-CoV-2 with the ACE2 receptors expressed in the blood vessels and various tissues and organs cause an infection. The infection causes an injury to the vascular wall of blood vessels, initiating coagulation. The final result of coagulation is the formation of blood clots in various organs, especially lungs. And I repeat, this is one of the expected things. Like RNA virus, coronaviruses have a high rate of mutations due to the lack of proofreading nature of RNA polymerase and all of them.
Dr. Najla, sorry, you'll have to come closer to your microphone. We lost
Shall
you
I
again.
Yes.
If you could stay closer to your microphone, please.
Yes, sure. You want me to repeat about the clots?
Oh, that's right, yeah.
Okay.
There was quite a bit of blank in the whole thing.
Yep. Patients with COVID-19 are at significantly increased risk for the formation of blood clots. The pathogenesis for their development is incompletely understood. One possible explanation is as follows. The binding of the spike protein of SARS-CoV-2 with ACE2 receptors expressed in the blood vessels and various tissues and organs cause the infection. The infection caused an injury to the vascular wall of blood vessels, initiating coagulation. The final result of coagulation is the formation of blood clots in various organs, especially lungs. Like RNA virus, coronaviruses have a high rate of mutation due to the lack of proofreading nature of RNA polymerase in all of them. So to know the differences, we have to know the sequences at first place. Researchers all over the world have sequenced different viral genomes. They have done phylogenetic relationship among different virus strains, which will indicate the track of how the virus is evolved. They also correlate sequence variation in viral RNA to disease severity and transmission efficiency. As we can see in this current slide, the main protocol of sequence the whole genome of SARS-CoV-2 virus, beginning with taking nasal swab as an example for COVID-19 patients, going through the validation by real-time PCR to complete the sequencing process, as it shows in this photo. This is in a clinical course. By January 2020, the whole genome sequence of SARS-CoV-2 was published. Like other coronavirus, the genome of SARS-CoV-2 is composed of single positive stranded RNA, ready for translation and synthesis of its proteins. The genome is considered large, with 29,903 base pair, which is considered to be really very big and large. As we see in the photo, it has two open reading frames, ORF1A and ORF1P, spike protein gene, and many other genes, and many other protein genes, actually. The high mutation rate of SARS-CoV-2 as RNA virus caused the emergence of new waves that affect the virus severity and transmissibility. Here we are represent the known virus mutation of waves regarding to some countries till now, of course. The explosive nature of COVID-19 transmission is initially shown by the number of new cases and later by admission and deaths. The principal worrisome about COVID-19 transmission are the mode of transmission, the role of asymptomatic infected people, its speed, the possible interaction with wildlife or livestock, city or country environments, and population density. Due to its explosive transmission and circulation, about 5 main waves have been happening. They are UK, South Africa, Brazil, California, and India waves. We'll talk about each of them. In UK, a variant with 17 mutation was detected. No increased disease severity, but increased risk of death, detected in more than 40 countries. In South Africa, a variant with 1 mutation was detected, linked to higher viral load and increased transmission, detected in 6 other countries. In Brazil, a variant with 1 mutation was detected, increased transmissibility, and potential for reinfection. In California, 2 variants were detected, linked to increased infectivity, and new research suggests that they may be more transmissible. In India, a variant with 2 mutations was detected, linked to increased transmission with mortality rate of 1% till now, detected in many other countries. The spike protein of SARS-CoV-2 and its mutation are very important topic to discuss. And it's very important, the spike protein, actually. As mentioned above, the presence of spike protein, it allows the virus to penetrate host cell, causing infection. The spike protein is attracted by some vaccine that currently being developed, and mutation of the spike protein are therefore closely watched. At present, the implication of the identified changes regarding to spike protein gene in the SARS-CoV-2 variants are not well understood and are being investigated. This picture shows the whole coronavirus particle with yellow color spike protein. And we can see the shape of it's like a crown, really. And there is the RNA gene and protein and a lot of protein inside. This is, it's general, in general. By now, we are reaching one of the most important parts of our talk, which is vaccines. In general, vaccination is the administration of a vaccine to help the immune system develop protection against a disease. There are many types of vaccines for COVID-19. All of them are trying to achieve the same things: immunity to the virus, stopping the transmission. Vaccines stimulate the immune response to a molecule found on the virus known as antigen. In the case of COVID-19, the antigen is typically the spike protein found on the surface of the virus, which help the virus to invade human cells. This figure, for example, describes the natural immunity against the virus. When the virus infects lungs or intestines cells, it causes tissue damage and inflammation, which induce the production of cytokine storm. After its replication inside the cell, it produces its antigens, which with cytokinase induce cellular and humoral immunity. So, to achieve this process, different types of COVID-19 vaccine have been made. There are 4 main types of vaccines in clinical use: viral vector, nucleic acid, DNA or RNA, protein subunit, and the whole virus. We'll talk about each one so we know the difference between all of them and the target of this vaccine. Viral vector vaccine work by giving cell genetic instructions to produce antigens, but they are different from nucleic acid vaccines in that they use a harmless virus. They are adenoviruses, which cause the common cold. Viral vector vaccine can mimic natural viral infection and should therefore, they should trigger a strong immune response as like AstraZeneca vaccine. Nucleic acid vaccines use genetic material, either RNA or DNA, to provide a human cell with the instructions to make the antigen. In the case of COVID-19, this is usually the virus spike protein. Once this genetic material gets into a human cells, it uses our cells' protein factors to make the antigen in large quantities that strongly trigger an immune response. RNA vaccines need to be kept lower as Pfizer-BioNTech vaccine, and Moderna is an example of DNA vaccine. So you can see the difference between two of them. Another type is protein subunit vaccines. Use often fragments of the protein to trigger an immune response. Doing so minimizes the risk of side effects, but it's also mean the immune response may be weaker. An example of an existing subunit vaccine is the Philander University vaccine, COVAX. Now we're talking now about the live attenuated vaccine and killed vaccine. Live attenuated vaccine use a weakened form of the virus that can still replicate without causing illness. It might cause diseases in people with weak immune system and often require carefully cold storage, such as codagenics, which is in a clinical trial right now. Live attenuated vaccine are unlikely to progress due to the difficulties in production and attenuating the virus. The killed vaccine use viruses whose genetic material has been destroyed, so they cannot replicate, but can still trigger an immune response. These vaccines can be given to people with compromised immune systems, but might also need cold storage as like Bharat Biotech, like Sinovac, and Sinopharm also, it's the same, yes. A nanoparticle-based vaccine, the pathogenesis DNA or protein is attached to a nanoparticle. Novax is an example of protein subunit vaccine. It's contain SARS-CoV-2 spike protein. Here, according to COVID-19 vaccine tracker, COVID-19 vaccine development has progressed quickly over the last year. Dozens of vaccine candidates are being tested in a clinical trial to evaluate their safety and efficacy. The landscape is changing rapidly with many countries now approved COVID-19 vaccine and launching vaccination campaigns. By June 15th, 2029, there were 123 vaccines candidates, 359 in trials, vaccine, and 17 approved vaccines. I think it's in this slide, I think it's better to follow the link for a new update. Let's see it, let's open it. As we can see, the numbers have been changed by now. Now there is a difference, there is 124 vaccine candidates and 361 on trial and still 17 approved vaccine. So if you like to update yourself, you can go to the link always. The efficiency of some COVID-19 vaccines is the information that we are all interested to know. This table shows some vaccine types, dose, and efficacy. For example, AstraZeneca is viral vector, which is a genetically modified virus vaccine with 2 doses. The efficiency rate are 10% against South Africa strain and from 62 to 90% against original strain. Pfizer-BioNTech is an RNA vaccine with 2 doses. The efficiency rate is 95% against original strain. Johnson & Johnson is a viral vector, which is also genetically modified virus vaccine with only 1 dose. The efficiency rate is 75% against South Africa and 72% against the original strain. Novavax is a nanoparticles-based protein subunit vaccine with 2 doses. The efficiency rate is 55% against South Africa and 86% against UK and 96% against the original strain. Sinopharm is an activated SARS-CoV-2 vaccine with 2 doses. The efficiency rate is 79% against the original strain. I give you some sample. I don't have to read it all. This is give you an idea about the type, the dose, and the efficiency, which is, it gives you really a right information. The current slide, this one, gives us example of approved vaccines in some countries representing the main 6 continents. For example, in the Kingdom of Saudi Arabia, AstraZeneca, Moderna, Pfizer, BioNTech, and Johnson & Johnson, so they approved 4 vaccines. In India, they approved 5 vaccines, which is AstraZeneca, Bharat BioNTech, COVAX, Sputnik 5, and Covishield. In Germany, AstraZeneca, Pfizer, BioNTech, Moderna, Johnson & Johnson. I think most of them, they have almost the same, but this is a reference. You can see all the continents, what they use. I pick one country from each of them as an example. But I want to mention in India, it's coming soon to India, Pfizer and Moderna also. So the approved vaccine, it will be 1, 2, 7 almost. By the way, the worldwide number of vaccinated people is 2.26 billion, representing 28.29%. On the other hand, the number of vaccinated people in KSA is 15.8 million, representing 46.21%, meaning almost half of the KSA population have been vaccinated till now, which is a good number. Maybe the end of this year, all population will be vaccinated. Here we come to talk about the vaccination of important society groups. The current slide presented the possibility of using COVID-19 vaccines in pregnant women, children, and people with chronic diseases. Pregnant women are more likely to get severely ill with COVID-19 compared to non-pregnant women. If you are pregnant, you can receive COVID-19 vaccine. Getting the vaccine during pregnancy can protect you from severe illness. Therefore, the American College, ACOG, recommended that pregnant women to be vaccinated. For children, it's a routine for children and widely accepted for different infectious disease. On May 10, 2021, the USA Food and Drugs Administration, FDA, expanded its emergency use authorization, EUA, for Pfizer-BioNTech COVID-19 vaccine to include children aged from 12 to 15. By Tuesday, June 8, 2021, Pfizer announced it's moving forward with the final phase of clinical trial for its COVID-19 vaccine, meaning kids as young as 5 are one step closer to being able to be vaccinated. For people who have chronic disease and illness, such as diabetes or heart problems, blood pressure, and also organ transplant, are at a high risk of serious illness and death due to COVID-19 infections. Therefore, they recommended to be vaccinated against COVID-19 according to WHO. And now what's next? Events since the time of the outbreak of COVID-19 are at high speed. In months, new mutations have been discovered. In the upcoming time, it might be proceeded and might be not. Treatment and vaccines are being discovered and development very quickly to combat this epidemic. And all kinds of precautions must be followed to reduce its speed, such as cleanliness, physical distancing, wearing masks, and taking the vaccine, each according to the country where he or she lives. For the future, and after the huge amount of information that we are already know about the nature of this virus till now, I think it will be easier and encouraging, either to eliminate the virus or live with it. So I'm very optimistic for the coming future. Finally, on my own behalf and on behalf of the citizens and residents of Kingdom of Saudi Arabia, I'm taking this opportunity to thank the custodian of 2 holy mosques, King Salman bin Abdulaziz and the Crown Prince Mohammed bin Salman bin Abdulaziz, who favored the citizens and residents' health in the first place. Thanks would also extend to the Minister of Health, Dr. Tawfiq Fahad al-Rabi'a, who went through a challenging experiment with the distinction that is being studied worldwide. Since then, strict health laws are being enforced in the kingdom. In addition, vaccines were made available to all citizens and residents without any compensation. Thank you. Thank you very much for your listening, and I wish you all happy life with good health without COVID-19. Thank you.
Thank you very much, Dr. Najla. That was a good session for me to get a perspective in terms of how the virus works and causes all the damage, and how the scientific community is coming together to fight it.
Thank you very much.
Thank you so much for this.
Thank you.
So we have started getting a couple of questions already.
Okay.
To the audience, if you still want to raise your questions, please do so in the Q&A box. And we have another 18 minutes with us, so I'll raise these questions to Dr. Najla. The first one, which is an audience choice question, is what is your opinion about the vaccine hesitancy, and how can we change this perception in people who are hesitant to get the vaccine?
Sorry, I don't hear you very good. What is what?
There are a lot of people who don't want to get the vaccines.
They don't want to get vaccinated.
Yes, because they are hesitant. So what would you like to tell such people?
The vaccine must be proven safe and effective in large, in large quantities. So many people that already take the vaccine, they were hesitated before, as they are the people who are asking. So many people, they were hesitating not to take this vaccine, but actually it's proved that it's safety, and it proved that it's protect them from getting the infection, which sometimes lead to a severe symptoms. So COVID-19 vaccine can help protect your child from getting COVID-19, can protect your family, your mother, your father, your grandfather, your neighborhood. So I think they have to think more and encourage themselves to take it, because we all take it and it's safe.
Right. So it's not only for us, it's also for the people around us and the society.
Exactly. Your friend, your father, your mother, your children, your neighborhood, your friends.
Okay. Wonderful. I hope that's a message that people can take away. Think beyond ourselves.
Because if you love your parents, for example, if you have parents and they are old, over 60 or 70, you have to protect them. You have to feel a fear that you want to keep them safe. So you have to think more than one time. You have to do it.
Wonderful. So the second question is sort of continuing on the vaccine thing. This is a question from Srihari. I'm combining two questions together. The human body has 120 trillion non-pathogenic beneficial bacteria. And why can't our natural immune system fight the virus? That is the question. Is it not strong enough to fight the virus?
He means sometimes, yes. Sometimes.
But not enough.
Because this is we explained before. That's why there is some people, they don't have any symptoms, without any symptoms.
Right. So this one is from, so there are a few people who are asking for the presentation. You will get a copy of it. We will publish it on our website, and we will let you know. So in case I'm just ignoring all those questions. Okay, here is another question from Vijay Kumar, which is about, there is a concern about the efficacy of the vaccine against the mutations. So are we seeing a future where the virus will keep mutating and we have to take different vaccines?
This is I think in this slide, I go back to it. This is the efficiency of the vaccines, and this is different. If you can, for different, there is different variants for the vaccine, and it's written here. For example, you can compromise, or he can see it and look at it. For example, Pfizer, it's 95% against the original strain. And for example, the Novax and AstraZeneca, all it's written. This is the efficiency of all the vaccines that it's already approved, I mean.
Okay. The next question is how SARS-CoV-2, how is it different from MERS and SARS clinically?
Okay, we go back to the slide. It shows, it's a typical science. That's why I don't go that deep here. This is the difference in the genome. In the UTR area, untranslated region, this is how can we compare between it. And we can see the SL4 prime stem loop. This is the difference between 2003 and 2019. Also, there is a difference in the receptors in SARS-CoV and SARS-CoV-19. The receptors special, it's named ACE2. It's different from the MERS-CoV, which is DPP4.
All right. I hope the person who asked the question understood it.
If he doesn't understand or he wants more explanation, I can email him. He can put his question and I give him all the details.
Wonderful. That'll help. So I'm going to stay away from this, and I'll take a simpler one. So this is from Mohammed Subin Siyas. The question is live attenuated viral vaccine better than the other types? If yes, why is Covishield given in India over Covaxin?
Well, it depends. It has to be safe before they approve it, or that's why it's good. Of course, it's not bad, but it has to be safe. They have to be tested. They have to be go on so many trials, and they approve it after all.
Okay, Ashima Rohila, the question is, what should be an ideal gap of duration between the two doses of vaccine, the first one and the second one? Maybe if I can add my perspective onto it, Dr. Najla.
It depends.
The gap kept changing. Initially, it was a few weeks, and then now it has become a few months. Why is that happening? On what basis is that being decided?
I'll answer the first question. It depends on the vaccine types, the first one. The other question, I don't get you right. What do you mean? I don't understand.
Oh, in India, first, I think it was 2 weeks or 3 weeks of gap between the first dose and the second dose. Then it became 12 weeks, and now it's about, I think, 84 days or so. So it kept changing.
Okay. The manufacturers who decide, the manufacturers who decide of WHO, I mean, the manufacturers, WHO,
Okay.
the company who decide to WHO.
It's based on the trials they decide and how.
Yes.
Okay, got it. Noof Zari, I hope I'm getting this name right. His question or her question is, could you please explain him?
Her question.
Okay. So, could you please explain the severe health complications and death cases after being vaccinated?
You mean the severe, somebody get affected, then after he recover, he want to vaccinate it? This is the question?
No, it is after being vaccinated, some people are still getting severe health complication and even ending up in death in certain situations. Why is that happening?
No, 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 no.
Okay. Vinny Lokhande, the question is, should we take any test?
Wait a minute. I think she wants to know what is the symptoms, yes?
Why is the symptom even after the vaccine? That is the question.
A side effect, you mean? Side effect? Or
Not
the
the
system?
side effect, infection. After vaccine, some people still get infected. Why?
Yeah, because it happens, of course, because some people maybe they don't know what they have maybe they do have kind of disease they don't know about it. So they discover after the vaccinate, maybe some complication happens, you know. The mortality rate after 2 doses or they get infection of COVID-19 after vaccine, they are very little anyway. But it's depend on the immunity of each person. I think the mortality is these cases for weakness of immune system and depend on the other different illness factors, you know. This is normal. I mean, it happens in many cases, and this is the only explanation. That's it.
Thank you, Dr. Najla. We have a few more minutes. I'll take up a couple of questions.
Yes,
This is
sir.
from Daniela Marie Segued. The question is about the mutation of the virus. She wants to know, aside from the environmental factors, what are the other factors responsible for the virus mutating? Is there any other reason apart from environmental factor?
Yeah, I mentioned it. I think it's that. No, just a minute. I think it's. I announced it before. I told you, it's depends of how is the environment, the capacity, the density, the city, the population, the cleanness, the awareness about the support the immune system. And it's like RNA viruses, coronaviruses have a high rate of mutation due to the lack of proofreading nature of RNA polymerase in all of them. So this is a good reason, or so many reasons, as I told you before. It's like.
Right. So there are quite a bit of things that come into picture. Another popular question, this is from Dr. Sameer Agarwal. A bit technical. What do you think about neutralizing antibodies testing?
Neutralizing?
What do you think about neutralizing antibodies testing?
Antibody test? Okay. They use it, I think, in them. If I get you right, the question, they use it in hospitals. They do this test. It's different from real-time PCR. This is different test.
Yeah. So the question is also,
they use it.
A booster dose? Sorry, please go ahead.
Is it what?
Should we test to figure out a booster dose, the anti-spike IgG antibodies? Okay, let me take.
I don't get the question right, really. That's why I cannot answer.
We can take it offline, Dr. Najla. I'll send these questions to you. We can take it offline.
Because it's not clear, the question. Otherwise, I can answer her. It doesn't matter. But I will answer her an email, everything she wants to know.
Perfect. Thank you. Okay, this is from Kumar Kapavarapu. His question is, if someone is already infected with COVID-19 from, let's say, mild to medium symptoms, would you advise them to take the vaccine?
Yes, of course. Yes.
So even if they have a natural immunity from the infection, they still need to take the vaccine.
Yes, after the symptoms goes and disappear, yes, he can take the vaccine, of course.
Okay, so this. Okay, the next question is, how vulnerable are infants, that is, really young children, to the current variants of COVID-19?
Children,
Infants, babies and infants.
infants. Ah, the children, you mean the children, yes?
Babies, yes.
Yes. Not babies, not yet. No, not yet. I mentioned that Pfizer announced 3 days ago they are almost in the trial to approve a vaccine for children in 5 years, but babies for nothing come yet.
Right.
For 12 and 15, for 12 and 15, it's already approved, but for the children 6, 5, it will be soon.
Okay. This is a very interesting question from Deepak. Would you recommend mixing the vaccines, a cocktail of sorts?
Yes, it happens now, and I know so many people, they do have different doses. Like, for example, some of them, they took AstraZeneca. I'm talking about what in Saudi Arabia. I don't know about other countries. But yes, some of them, they took AstraZeneca, and the second dose, they took Pfizer. It's I don't think there's any problem. Maybe even though to make the immune system strongly.
Maybe. Guess there is quite a bit of around
Yes.
that.
Yes.
I'll take one last question. I'm not sure if this is the right forum to ask, but anyway, I'll just ask. This is from Vijdan Abu Turkia.
Yes.
What is your view for chloroquine or hydroxychloroquine as a treatment for COVID-19?
You mean the antiviral?
HCQ, the
Yeah,
HCQ antiviral.
yeah, the antiviral. What's the name of the medicine or the antiviral?
Hydroxychloroquine and chloroquine, HCQ.
Yes, I understand now. Yes, they tried before. They said it's good and it helps the immune system, and it might be a good antiviral, but it's good, but the vaccine is better than the antiviral. We don't, because there is some mysteries about this COVID-19. We don't know it yet. So as we know, so many waves coming, that means it's changing. The mutations change every time, and rapidly. So the antiviral might change also. Maybe it's covered this mutation, but doesn't cover the other mutation of the changing in the virus. The vaccine, it's more safe, I think, and give you the really purpose that you want. I mean, protect yourself from COVID. So it's okay, but it's not as strong as the vaccines. This is a different story between them. This is antivirus and this is vaccine.
So prevention is better than treatment, yeah?
Yes.
Okay,
yes.
Wonderful.
Yes.
With that, we'll bring this to a close. The time is up. I wish we could hear a little more, but we'll take all these unanswered questions offline and we'll publish it on our website. And meantime, if you have any further questions for Dr. Najla, please email it to me, vijay@kivani.in, and I'll pass it on to her. A copy of this presentation and a recording of this webinar will be available on our website. So just watch our social media channels. You'll get to know when it is available. With that said, thank you, Dr. Najla. It was
thank you very much. Thank you very much.
A great pleasure to have you and hear your views. And thank you for sharing your knowledge with us.
I hope it's good for everyone. Inshallah, I hope so.
It was, given the amount of misinformation that was happening, it's good to have an expert view as to what is right and what is not. And to that extent, this clarified quite a bit of confusions that I personally had in my mind. So thank you so much.
Thank you.
Yeah. So with that, thank you everyone for making time to join us today. We'll see you in the next webinar. Until then, this is Perspectives, a thought leader initiative by Kiwani. Thank you and bye-bye.